have the opportunity to get to know many members of the Cancer Center community, and I'm looking forward to getting to know many more of you as well. So we're starting up a research effort, lab-based program here at Yale, and so today I'll be talking to you about one of the projects that we're focusing on here at the beginning. And as Roy mentioned, in addition to this, I'm also a member of the lung cancer group here at Yale, helping to take care of our folks with lung cancer at Smilo. So these are my disclosures. So today, part of what I'll be talking about today includes some work for which research funding support has been provided by Agios. So I've been broadly interested in understanding and using functional genomic approaches to try to help us address some of the challenges and limitations associated with the use of targeted therapies in cancer. So for instance, in one project, we've leveraged an overexpression functional genomic approach to identify and characterize genetic drivers of resistance to targeted therapies in lung cancer. And in a separate project, we've used similar approaches to identify and characterize a novel cancer vulnerability that we hope can be exploited for therapeutic purposes. And this is the project that I'll actually be focusing on today. So in lung cancer, really in the last 10 years, there's been a lot of progress in identifying targetable oncogenic drivers. So these are genes in which we can identify mutations or alterations that basically lead to constitutive activation of the proteins encoded by these genes that basically activate pathways that promote cell survival and proliferation. And so a number of these oncogenic drivers are shown here. And in parallel with that, there's also been a lot of progress in developing small molecule inhibitors to target these oncogenic drivers and to shut down those pro-survival and proliferative pathways. But despite the fact that a number of these targeted therapies can have oftentimes dramatic activity in treating patients whose tumors harbor activating alterations in these genes, unfortunately, we really tend to find these targetable oncogenic drivers really only in a minority of patients. So we really aren't able to use targeted therapies for most of the patients that we see who have cancer, both lung and other cancers as well. And in fact now, with data from large-scale genome discovery efforts, such as from the TCGA, with sequencing of hundreds of lung and other cancers, at this point we've likely found most, if not all, of the targetable oncogenic drivers. And so one approach that can be taken to try to identify additional potentially actionable genetic alterations is to exploit so-called synthetic lethal cancer dependencies. So these are vulnerabilities, cancer vulnerabilities that arise in the context of a cancer-specific genetic alteration. So for instance here, gene B becomes a potential therapeutic target only in the context of a cancer-specific genetic alteration in gene A.
Oh, we tried it multiple times, yeah.
And so this idea of trying to identify additional potentially targetable genetic vulnerabilities has actually been a focus of interest in the cancer program at the Broad Institute, which is where I had done my postdoc. And so one of the approaches that's been taken there to try to identify additional genetic vulnerabilities in cancers has been to try to integrate insights from two ongoing large-scale projects at the Broad. So one of these is called Project Achilles. This is a project in which hundreds of cancer cell lines have been subjected to genome-scale loss-of-function screening to try to identify novel cancer vulnerabilities in profiled cell lines. And then in a separate project called the Cancer Cell Line Encyclopedia, or CCLE, here hundreds of cancer cell lines have been subjected to comprehensive genetic characterization. And so the idea is that by integrating functional insights from Project Achilles with genomic insights from the CCLE, that it may be possible to identify new cancer vulnerabilities that are associated with specific genetic features. And so in analyzing data sets generated from these ongoing efforts, one of my colleagues at the Broad, a talented computational biologist, made an interesting observation. And what he found is that there were two genes, PRMT5 and WDR77, whose expression was essential for survival of cancer cell lines with homozygous deletion of a gene called MTAP. So in other words, genetic depletion of PRMT5 or WDR77 with RNA interference selectively impaired the viability of MTAP-deleted cancer cell lines, but not MTAP-expressing cancer cell lines. And importantly, actually the protein products encoded by these two genes, it turns out, are known to associate with one another in a functional complex called the methylosome. So here, PRMT5 acts as a catalytic core of this complex and serves as an arginine methyltransferase, meaning it transfers methyl groups to the arginine side chains of target proteins. And it turns out that PRMT5 is one of 9 known mammalian protein arginine methyltransferases, and collectively, this family of proteins has been implicated in the regulation of diverse biological processes, as shown here. PRMT5 in particular has been implicated in methylation of various histone proteins, suggesting a role in transcriptional regulation, and various spliceosomal proteins, suggesting a role in RNA processing. And WDR77, as I mentioned, is a binding partner for PRMT5. It's thought to function as a modulator of PRMT5 activity. And then importantly, S-adenosylmethionine, or SAM, is a critical cofactor that's required for activity of these PRMT family members. So SAM provides the methyl group that is used by PRMT family members for the methyltransferase reaction. So that's a little bit of background about PRMT5 and WDR77, but what about MTAP? So this is the gene that, when deleted, confers this dependency on the PRMT5 complex. So MTAP, it turns out, is a ubiquitously expressed gene in normal tissues, but it is frequently deleted in cancer. So for instance, MTAP loss is observed in over 40% of glioblastomas, in about 25% of esophageal, bladder, and pancreatic cancers, melanoma, and about 10 to 15% of non-small cell lung cancer. So it raises the question of why is MTAP so frequently deleted in so many different cancers? And the reason for this is due to its chromosomal proximity to CDKN2A, which is one of the most commonly deleted tumor suppressor genes in cancer. So when MTAP is deleted, it is nearly always co-deleted with CDKN2A, and we think that it's lost as a passenger event in the setting of CDKN2A loss. But despite this association between MTAP and CDKN2A, the genetic data from Project Achilles really seem to suggest that it was in fact MTAP and not CDKN2A that is the genetic feature associated with this dependency on PRMT5. Because as you can see here, it's really the cancer cell lines with loss of both MTAP and CDKN2A, shown in red, that demonstrate this increased sensitivity to depletion of PRMT5 or WDR77. Cancer cell lines with loss of CDKN2A but not MTAP, shown in the middle column here, do not demonstrate this increased sensitivity. And furthermore, this differential sensitivity between MTAP expressing and MTAP deleted cancer cell lines is present across different cancer lineages, so really underscoring the generalizability of this finding. And so we set out first to confirm that MTAP, in fact, does have a role in mediating differential sensitivity to knockdown or depletion of these two genes. We did this by choosing 4 MTAP-deleted cancer cell lines, so 2 non-small cell lung cancer lines, 1 glioblastoma line, and 1 pancreatic cancer line. And so for each of these 4 MTAP-deleted lines, we ectopically expressed MTAP. So we created an isogenic sister cell line with ectopic MTAP expression. So now we have 4 isogenic cell line pairs, with the only difference between the 2 members of each pair being the presence or absence of MTAP. And so we could then use these tools to ask whether the presence or absence of MTAP modulated sensitivity of these cell lines to knockdown of PRMT5 or WDR77. And that's what's summarized here. So basically, what we observed is that there was a partial rescue of sensitivity to depletion of PRMT5 or WDR77 with MTAP reconstitution shown here in blue, so suggesting a role for MTAP in mediating differential sensitivity to knockdown of these genes. So we were also interested in trying to understand what's the mechanistic basis of this association between MTAP loss and dependency on PRMT5. So it turns out that the MTAP gene encodes an enzyme that cleaves a metabolite called methylthioadenosine, or MTA, to generate precursor substrates for adenine and methionine salvage pathways. And it turns out that, interestingly, in the literature, there were reports that MTA can inhibit the catalytic activity of PRMT family members. And the reason for this is because MTA is actually a structural analog of SAM, the methyl donor required for PRMT5 and other PRMT family member activity. And so it turns out that MTA and SAM can actually compete with one another for binding to PRMT family members. And so this led us to speculate or to hypothesize that what might be happening here is that in the setting of MTAP loss, that could lead to increased intracellular levels of MTA, which in turn could inhibit the catalytic activity of PRMT5. And since PRMT5 activity appears to be essential in most cell types, these MTAP-deleted cells may have some degree of increased basal inhibition of PRMT5 due to excess MTA, thus making these cells uniquely susceptible to further reduction in PRMT5 activity with RNA interference-mediated gene depletion. And so we set out to test this mechanistic hypothesis. And the first component of the hypothesis is that loss of MTAP can lead to increased intracellular MTA levels. So to address this, we worked in collaboration with the metabolite profiling platform at the Broad to develop a mass spectrometry-based assay for quantitating intracellular abundance of several dozen metabolites, including MTA. We did this in our 4 isogenic cell line pairs, so basically looking to see whether we see significant differences in levels of these metabolites depending on whether MTAP is present or absent. And from looking at the heat map, what you can see is that for most metabolites, it really didn't matter whether MTAP was present or absent. Levels of most metabolites were not altered significantly. However, for MTA, you can see that actually in all 4 profiled isogenic cell line pairs, there was anywhere from a 1.5 to 6-fold reduction in relative abundance of intracellular MTA. We then asked more generally, well, if we just look at cohorts of MTAP-expressing and MTAP-deleted cancer cell lines from various lineages, do we see differences in MTA levels in these 2 cohorts? And in fact, we did see about a 4-fold increase in intracellular MTA levels in our MTAP-deleted lines compared to MTAP-expressing. So our conclusion from these experiments was that MTAP loss is in fact associated with increased intracellular MTA levels. So the second component of our hypothesis was that MTA can inhibit the catalytic activity of PRMT5. And we set out to investigate this using a commercially available radioisotope filter binding assay. So here, essentially, we queried the ability of MTA to inhibit the catalytic activity of a panel of 30 different methyltransferases, including several PRMT family members. And basically, of the 30 methyltransferases profiled, PRMT5 was the one whose activity was most significantly impaired by MTA. This occurred at nanomolar concentrations of MTA and with about 100-fold selectivity over the next most sensitive methyltransferase profile. And so while the literature had suggested that MTA was actually a general inhibitor of PRMT family members, our interpretation of this data was that MTA is actually a selective inhibitor of PRMT5, which is actually consistent with the data from Project Achilles, because it was PRMT5 selectively that was identified as a dependency, not other PRMT family members. And so, others have also been working on this, and work from other groups has shown that this dependency of MTAP-deleted cancer cells on PRMT5 and WDR77 actually extends to include additional members of the PRMT5 pathway. So for instance, this gene encodes an enzyme MAT2A, which is the enzyme that generates SAM, that critical cofactor required for PRMT5 activity. So like PRMT5 and WDR77, MAT2A, when depleted with RNA interference, selectively impairs the viability of MTAP-deleted compared to MTAP-expressing cancer cell lines. So obviously there's been interest, just given how common MTAP loss is in different cancers, in trying to develop therapeutic strategies to try to exploit this potential vulnerability for therapeutic purposes. And while to date, although there are PRMT5 inhibitors, unfortunately, in the setting of excess MTA due to MTAP loss, most of our existing PRMT5 inhibitors really have not proven effective in terms of impairing cell viability. However, recently, a first-in-class oral MAT2A inhibitor has been developed by Agios. And so this potentially could provide a strategy of indirectly impairing PRMT5 activity by targeting MAT2A. So this is some data I'm going to show you from Agios. This was presented at a Keystone talk last year, and this is data that's publicly available on their website. So this is just some characterization of their oral MAT2A inhibitor. So basically here, what you can see is that their compound is associated with a dose-dependent reduction in intracellular SAM levels, so consistent with on-target activity against MAT2A, which generates SAM. And then here you can see that with their inhibitor, that there is selective activity against an MTAP-deleted cancer cell line compared to MTAP-expressing. And in fact, when they profile their compound across over 300 cell lines, you can see that MTAP loss actually predicts sensitivity to this compound, as would be predicted from the genetic data. And then finally, here, their compound impairs the growth of an MTAP-deleted mouse xenograft model system. So collectively, these findings suggest a potential therapeutic strategy of trying to target PRMT5 in the setting of MTAP loss by targeting MAT2A. So there's some degree of basal inhibition of PRMT5 due to MTAP loss mediated by excess MTA. Now, if on top of that, if we can inhibit MAT2A, reduce SAM levels, can we now even further inhibit PRMT5 and have activity in patients? So these are the preclinical findings have now motivated this strategy, which is now being enacted in a phase 1 clinical trial, which we hope to have open here at Yale within the next month. So this is a trial, this is a first-in-human trial of Agios' MAT2A inhibitor. And patients basically who have advanced solid tumors or lymphomas with homozygous loss of MTAP will be eligible for enrollment. So MTAP loss will be established centrally by the sponsor by an immunohistochemistry assay. But importantly, since about 60 to 70% of cancers with homozygous loss of CDK and 2A, since about 60 to 70% of those also have homozygous loss of MTAP, patients with loss of both copies of CDK and 2A will also be eligible for enrollment. Agios will be doing central immunohistochemistry testing to confirm loss of MTAP. And so CDK and 2A loss can often be identified on our currently available next-generation sequencing platform. So hopefully we'll be able to readily identify these patients. So obviously we're looking forward to being able to participate on this trial. Again, hopefully it'll be open in a couple of weeks here. But in addition to, so in parallel with the trial and sort of getting back to the lab, we're also interested in kind of going back to our functional genomic toolbox to try to think about approaches that we can use to understand what the consequences are of inhibiting the PRMT5 pathway in MTAP-deleted contexts. So specifically, we'd like to be able to try to identify genes and pathways that have the potential to rescue viability of cells exposed to PRMT5 pathway inhibition, such as the MAT2A inhibitor. We think this work could be important in helping us to identify downstream effector pathways regulated by PRMT5 that could be important for maintaining cell viability, and potentially to identify genes and pathways that, when activated, have the potential to compensate for PRMT5 pathway inhibition. So this could be informative in understanding potential mechanisms of resistance to targeting this pathway, and could also suggest additional potential therapeutic targets that might be incorporated as combination strategies for targeting PRMT5 pathway, or potentially as alternative if it seems that viable clinical candidates can't be developed for PRMT5 or MAT2A. And so, the approach that we're taking for this actually involves a modified CRISPR approach. So obviously, as everyone here knows, so with CRISPR, this is an approach in which via guide RNAs, double-strand DNA breaks can be introduced into targeted genes of interest. But we're looking at using a modified version of this approach called CRISPR-A, or CRISPR activation. So basically, this involves the use of a catalytically inactive Cas9 molecule. So this molecule can't introduce those double-strand breaks that ultimately leads to inactivation of those genes. Yet it still can recognize guide RNA sequences, so it can be directed to specific loci in the genome. And then the Cas9 is fused to a transcriptional activator, and the guide sequences that are used in this approach also contain binding sequences for additional transcriptional activators as well. So the idea here is that rather than a loss of function CRISPR screen, this is actually the opposite, a gain of function screen. And the idea is that by using guide RNAs directed to the promoter of a target gene, by sort of recruiting this transcriptional apparatus to basically get activation of endogenous genes. And now there are libraries available with guide RNAs directed to the promoters of nearly every gene in the genome. So with a screening approach like this allows you to basically systematically go through and try to upregulate each individual gene and to look for the activity or the phenotype for the effects of that upregulation on a particular phenotype. So essentially here we'd be taking the Broad's CRISPR-A guide RNA library, introducing them via lentiviral infection into an MTAP-deleted cancer cell line for which we have already introduced the Cas9, modified Cas9 system. And then we basically expose guide-expressing cells to the PRMT5 pathway inhibitor. The majority of cells would die, but then we characterize the cells that survived to determine what genes were upregulated that allowed the cells to circumvent sensitivity to the inhibitor. And so this is work that's being led by Lisa Hunahan here in the lab. And so this is data that she's generated. This is actually our first data generated by lab here at Yale. So here she has taken 3 different MTAP-deleted lung cancer cell lines. She's introduced the modified Cas9 into these, and then she's introduced either a control guide RNA or a guide RNA directed against the cell surface marker CD4. And so then using an antibody directed against CD4, what you can see is that for cells transduced with this guide, that we see a shift in the cell population corresponding to induction of cell surface expression of this molecule, findings that are absent using a control guide RNA. So Lisa's been able to now demonstrate with this that we can use this CRISPR-A system to induce activation of target genes in these cell line systems, and she's now working towards optimizing conditions for screening so that we can choose one or more of these to move forward with the screen. So we're really excited about being able to get started with that. So I think I'll stop there. I just want to thank obviously Lisa for all of her work in sort of helping the lab to get up and going and moving this project forward. To my prior mentor at Dana-Farber, Levi Garraway, Gregory and Jason also had leadership roles in this project. Folks in the Cancer Center who've been providing mentorship and guidance for me as I get started, and great collaborations with Agios and to the Phase 1 group as well for all their efforts in getting this trial up and running. Thanks very much.
A couple minutes for questions. So any folks? Jeff.
So when we sequence tumors, we much more often see point mutation in CDKN2A and deletion of the second allele. So I'm wondering whether you've investigated the status of MTAP in those cases. Is it methylated, for instance? So it may be deleted by the LOH, but there's still a gene there apparently in the second allele.
That's absolutely right. So you're really so far in terms of the profiling that's been done in terms of trying to identify tumors that have MTAP loss. Initially it was based on loss of both copies or loss of expression from RNA-seq, simply because that was sort of the cleanest way to be able to identify it at the DNA level. But you're absolutely right. You could certainly imagine a scenario where there's loss of function. So you lose one copy, maybe you have then a missense mutation or a nonsense mutation in the remaining copy. So those, it's a situation where you really need to have actually loss of both copies to have loss of CDKN2A so that you have loss of both copies of MTAP. But the other possibility is, as you're alluding to, is that there's also the possibility that you could have a non-DNA mediated mechanism of loss of MTAP expression. And in fact, there's some data to suggest that certainly with methylation of the promoter of MTAP, you can have repression of expression there. So it could be that we could be underestimating the frequency with which this is observed if we're only looking at a DNA-based assay, which is why I think Agios is also incorporating an immunohistochemistry assay as well to be able to look at this, which might be able to capture some of those.
So I have another question, just slightly related. So in your CRISPR activation of transcription, how sensitive is that to things like methylation, heterochromatin formation? Can you overcome those usual suppressive signals in DNA?
Yeah, I think there are some questions, associated with the technology. It's fairly new. It has been used in screens like this in the past to basically try to repeat screens that have been used with other gain-of-function screening technologies, and it has been observed that there's been overlap in those findings. But certainly you could imagine that it could be situations where if promoters for certain genes are not necessarily accessible to the machinery here, that you may not be able to induce expression of those. There's also a possibility that if you've already got some degree of high-level expression with some endogenous genes, you may not be able to augment it further. And the other thing that we don't know is whether, by inducing these endogenous expression of these genes, are you going to be able to maintain some of the different splicing isoforms that may be relevant, with endogenous expression that you may not be able to replicate in this way. So, there are some limitations like that you could certainly imagine in terms of being able to really be able to assay every gene for which you have guides in the library.
One other quick question.
I noticed you went from cell lines to humans. Are there no animal tumors that are MTAP deficient?
So I did sort of skip from that in the talk today. So there has been work looking at this primarily in xenografts. Mostly have been taking MTAP-deleted cancer cell lines and using those to create xenografts. Both then with knockdown in those situations, and also, as I showed one brief slide of, Agios using this with their compound. Certainly, I think it's a situation where, given how prevalent MTAP loss can be observed in different cancers, that it would be very interesting to look back at, take a look at existing PDXs, that may already have been generated for other purposes, particularly in certain cancer lineages in which we see higher prevalence of MTAP loss, to be able to see. I bet that we find that there's some of these models may have already been developed that we could certainly use for these types of studies to be able to look in vivo.
Well, thanks a bunch. Let's thank Rick for a nice talk.
Thanks.
And now we'll get our second speaker set up. And that will be Jun Lu. And I'm looking forward to Jun's talk. He's a collaborator as well, and making sure I get your title correct here.
Well, that was actually
Nothing to disclose tonight. The impact of clonal hematopoiesis on cancer. We're running a little short on time, so I'll let Jun start. Thanks.
Thanks, Marcus. Very loud. Yes, nothing to disclose. So it's actually really proper to talk about this here because Marcus is our very great collaborator, without whom we will never be able to do this line of work. So the talk will be about a phenomenon called clonal hematopoiesis. I think this is a term that's been very familiar in recent years by folks in the hematology field, especially if you're working with a normal hematopoiesis or malignant hematopoiesis. But I guess it hasn't been widely spread to every single cancer areas yet. So one of my job today is to tell you why this phenomenon that's actually occurs in blood cells, why actually may be not only important for blood cells cancers, but also for solid cancer as well. So what is clonal hematopoiesis? So this is actually a phenomenon that was initially described about 5 or 6 years ago, and it was really starting from work that looking at so-called the germline DNA that we have. So I bet many of you know that in many large-scale population studies, we use peripheral blood cells as germline controls, with the assumption that the germline cells present, the blood cells present in the bloodstreams are normal cells that reflect your germline genetic variations. But turns out that's not 100% the case. So if you look carefully at those samples with peripheral blood samples by deep sequencing, you actually find there are variations in the genetically that's not 100%, so it's a mosaic. And this is really termed as now termed clonal hematopoiesis. So the observation, if you look at the graph on the very left, is that among people with different aging populations, especially with ages over 60 or 70 years old, you have a clearly detectable at that time, so I want to emphasize at that time, level of mutations, somatic mutations within the peripheral blood samples. And the frequency allele, which you can look at on the other side, is roughly 0.1% allele frequency on average. So that means about 20%, if it's a heterozygous mutation, about 20% of blood cells are mutated on average, with the extremes will be approaching 50% allele frequency, which means 100% of cells are mutated if there's a heterozygous mutation. Now, one of the limitations, of course, with these studies is that sequencing depth was from established studies with 30x, roughly 30x genome coverage. So that means the sensitivity to detect rare mutations in those blood cells will be roughly 5% or more. So if you have potentially mutation levels, actually number of cells carrying mutation less than a few percent, you are not going to be able to see it. So that's a limitation of the time. But nevertheless, we start to appreciate that there are a huge group of people with not major symptoms in cancer or beyond actually will carry this mutation around. Now, so which genes are mutated? So the mutation doesn't occur in any single gene, but really occurs focused on a number of genes we actually have been knowing playing important roles as tumor suppressors in the blood cell system. And some of them are actually well known to play a role to control the copy of the cell numbers of hematopoietic stem cells when they're deleted. So the top two genes, I don't know if you can see here, one is DMP3A, which is here, and one is TET2. And both of them actually is biochemically in the same molecular pathway, which is to regulate methylation on the genomic DNA. And many of you are aware that the genomic DNA methylation levels control gene expression patterns. And paradoxically, a loss function of DMP3A, which is supposed to be doing methylation, and the loss of TET2, which is supposed to be demethylation, actually achieves the same effect on hematopoietic stem cells. Both will amplify hematopoietic stem cells. So what do these data really mean? So this is basically the model that we have now to use to interpret these data that we observe in human populations. Is that initially we have wild type normal stem cells, hematopoietic stem cells, in humans. And then at one point, a single clone of hematopoietic stem cells will acquire a mutation, and these mutations happen to be in one of those important genes that safeguards the genomes of the stem cells to prevent it from overamplification. And once you have this clone of a position of this mutation, this clone gets expanded, and it takes over the system gradually. And this really starts to lay the ground for further mutations to get eventually leukemia or other kinds of blood cancers in the course. So this is basically our interpretation of the the process, and initially we think it applies to about 10% of population older than 60 years of age. But now some recent studies really says this is actually not true. This is actually potentially a universal feature of aging, that almost everyone will get it beyond a certain age. So this is a study done recently, and looking at a very small scale study, about 20 nurses who are aged between 50 to 60 years of age. And looking at the 20 individuals in this study, 19 of them have clonal hematopoiesis hitting those important tumor suppressors. So really, if you thinking this is a representative of the general population, that roughly says that this is a universal feature of aging. And you can see the top two genes, one is DMD3A, one is TET2. So what's the so apparently it has something to do with blood cancers, right? So we know that these are the initial mutations that actually frequently occurred in human leukemia, for example, and other hematopoietic malignancies. So apparently, if you have a condition in the general population during aging process to acquire the very first mutation in the stem cells, you probably will have a higher chance to acquire later stages of malignancy. And the answer is yes. So if you look at the human population data as analyzed in this particular paper, there's roughly 5 to 10-fold increased risk to acquire some form of malignancies in the hematopoietic system. But I do want to point out, which I come back to, is that the lifetime risk is only about 4%. And this is comparing the early classification of clonal hematopoiesis in the field, which is something you can detect at their 30x coverage, roughly, versus something you cannot detect at 30x coverage. So effectively, this really means is that those individuals carrying higher allele frequency load versus those who are carrying low allele frequency load will have a difference in the risks to eventually develop hematopoietic malignancies, but yet only a small percentage of them will actually ever get any form of hematopoietic malignancy. So this really poses a number of questions in our own minds, and I think for the field as well. So one thing is, of course, we now know that this clonal hematopoiesis is now seems to be a general phenomenon of aging, and that many people, almost everyone, will live with some condition of this with expanded clone of hematopoiesis that carrying the mutations in their stem cells. And we know that, of course, one of the consequences when you have a stem cell mutation is that will be inherited genetically by the cells that give rise to. And very interestingly, in the blood system, the stem cell give rise to immune cells. So the question is whether the mutations acquired in the stem cells and once inherited by immune cells, will they have any impact on things beyond blood cancer, such as in solid cancer formation process or responses towards pathogens, which is by itself a very interesting process because many cancer patients are immunodeficient to some degree. They're more prone to infection and so on and so forth. So we started to look at this with the helper markers, and this is just to show that a section of a typical human melanoma section, you can see there are lots of, the brown cells stand here are macrophages, so there are lots of blood-derived cells within solid tumors. I think this is not a surprise for many of here who are pathologists. That's why we've been starting to focus on the myeloid cells in the process. So the first experiment we did is at the time when we were starting to think about these experiment, we heard about the story in Mark's lab where they developed a mouse melanoma model very similar to the human melanoma with BRAF, P10, and the P16 alterations, and then you can take a cell line and reproducibly generate melanoma in the mouse. So when we did that, injected the cell line into tumor, we simply asked the question whether or not the injection of tumor into the mice will change the expression of TET2, which is this gene involved in mutated in clonal hematopoiesis, the expression level for the myeloid cells within the tumor. And you can see here on the left is that indeed we can see that the tumor, the myeloid cells isolated from a tumor, in this case macrophages, have higher TET2 gene expression levels compared to macrophages isolated from other parts of the mouse body. Right? So suggesting that there's an effect by the tumor to reprogram the microenvironment to lead to an increased expression of TET2 within the tumor microenvironment, and compared to TET3, which is a family member which is not changed. So this is basically seeing what we're thinking is happening in the tumor cells, reprogramming the myeloid cells expression for TET2. And this is not only happens in mouse, but also happens in human. So we isolated myeloid cells from the tumor itself from melanoma patients versus the circulating blood from myeloid cells from the same patient. And you can see that there's increased TET2 detection in the tumor myeloid cells versus the circulating peripheral myeloid cells, suggesting this is a conserved regulation in human as well. So to address whether the TET2 regulation has anything to do with the tumor functionally, we decide to knock it out. So we can take TET2 flox mice, cross with a specific tree to delete specifically in the myeloid cells of the mice. So that the resulting mice will lose TET2 only in the mature myeloid lineages, and then we can start to challenge with the melanoma cell by itself actually is wild type for TET2, and to see what's the effect of host loss of TET2 in myeloid cells and the impact on melanoma development. And when we did that, we see that the tumor developed in the wild type cases are larger than those one developed in the knockout cases in the host. And this is not a huge effect, but it's a little bit surprising to me because, for me, TET2 is a tumor suppressor, and when you lose it, well, whatever cancer will get bigger, but in this case, tumor becomes smaller, which really suggests that this is a non-cell intrinsic sort of facilitating or promoting effect by TET2 on the tumor growth, in this case, solid cancer growth. So then the question is, what are the mechanisms that leading to these two phenomena? One is tumor cells reprogramming the myeloid cells within tumor to express higher level of TET2. Number two is TET2 within the tumor-associated myeloid cells to promote the size of the solid tumor progression. So I probably don't have time to show you all the data, but just to quickly show you some of the data. The first one we think is done by involving potentially the R1 receptor signaling pathway. Many of you probably know that the R1 receptor signaling pathway binds to ligands such as R1 beta or R1 alpha and signals to MyD8 and downstream to NF-κB pathway. And we think this is involved. I'll just show you some evidence. So this is, we take just some regular macrophages you can isolate from mouse and differentiate them and start to treat with a ligand for R1 receptor called R1 beta. And you can see after treatment of ligand, TET2 level goes up very dramatically, very quickly for the RNA level, protein level, as well as biochemical product level. So it seems to be, this is really a very quick response. And the R1 ligand is sufficient, R1 receptor ligand is sufficient to induce TET2 gene expression. To address whether R1 receptor signaling is required for the process, we decide to knock out R1 receptor in the mouse, and then start to challenge the mouse with the tumor cells, then isolate myeloid cells from the tumor to assess what's the level of TET2 gene expression. And when you do that, you can see that TET2 gene expression actually decreased in tumors grown from R1 receptor knockout. So actually in the myeloid cells purified from tumors that grow in the IL-1 knockout mice versus control mice. So indicating that IL-1 receptor signaling is required. Similar data can be obtained if we knock out MyD8 instead of IL-1 receptor, so really indicating this pathway is involved. Now, after that, then we have to turn to the second question: is how does the myeloid cells within the tumor, a tattoo within the myeloid cells in tumor promotes the tumor formation process? And I'll just show you very quickly the model and show you some piece of evidence supporting that. So our model is basically here: is that tumor cells through secreting interleukin-1 receptor ligands, upregulates the tattoo within the myeloid cells within the tumor. And the tattoo is very important to sustain the expression of genes that's important to suppress the adaptive immune system. And one of the major genes involved in here is a gene called arginase 1 or Arg1. And these immunosuppressive genes then in turn suppresses T cells within tumor, and these T cells are normally important to survey and eliminate solid tumors. And so basically tumor self-promotes with a double negative loop in the process. But if you knock out tattoo, what happens is that you have a decrease of this gene expression and decreased myelosuppressive effect of the myeloid cells, leading to increased T cell presence in the tumor, as well as these tumor cells, T cells actually functionally against the tumor. So I don't have time to show you every single piece of evidence, but just a few pieces. Number one is if you look at the wild type tumors growing in the wild type mice or tattoo knockout mice, you can see the arginase 1 expression in the myeloid cells purified tumor is dramatically reduced in the present tumor. So Arg1 is a very well known immunosuppressive gene, as well as a few other immunosuppressive genes. Number two is that, as we predicted, the level of T cells present in the tumor will be dramatically increased when you knock out tattoo in the myeloid cells, which is the case. So on the very left, you can see that on average in the wild type, we can detect roughly 20% of all the cells within the tumor are coming from the blood cell lineage, whereas in the tattoo conditional knockout ones, we see that that approaches 40%. In some extreme cases, more than 50% of whole tumor mass is made of blood cells. And the major increase here is due to the T cell increase, CD3 T cell increase, and they are actually mostly CD8 T cells increase we can see. So these are cytotoxic T cells. And then later we also proved that indeed these T cells are against tumor, and we also show that tattoo seems to be regulating this very important immunosuppressive gene called Arg1 through modulating methylation levels of Arg1's enhancer region. So what these data really tells us is that even though clonal hematopoiesis potentially is a phenomenon initially observed for the field of hematology, it may have actually much far-reaching implications in the cancer field beyond solid hematopoietic cancers. Right? So the first thing, this tumor-suppressed tattoo seems to have a non-cell intrinsic tumor promoting function, so promoting reprogramming the innate immune gene expression, and this could have a very widespread phenomena for clonal hematopoiesis, and should be potentially further investigated in solid cancers. So in the last few minutes, I want to share with you some of my thoughts along this area and some of the things we are working on, and some of the things are still puzzles for field that are potentially very, very interesting and worth further examining. So one of these questions clearly will be, there are lots of things need to be done to further clarify what's the role of these clonal hematopoiesis genes in the process. We worked on a single gene in a single cell type within the immune system. What happens with other genes? What happens with other cell type in the process? What if you have a mosaic condition versus complete knockout condition? What if you have a different single allele versus double allele losses? So these are all very important technical questions. But there are also other very interesting questions. For example, what's the relationship to immunotherapy? Are these clonal hematopoiesis relevant to the successes or the failures of immunotherapy? Can we see similar effects in different cancer types? Are there differences in cancer types that respond differently? Personally, I think that's the case, although we don't have data to 100% say that's true yet. And also, as I mentioned, is that not only against the tumor development process, these mutant immune program may also have a role in working against pathogens, and we know that pathogen infections are quite common in cancer patients due to various reasons. So that's one clear area that definitely needs more work on. The second area, I think it's also an extremely interesting process. Now we know that clonal hematopoiesis may have a very far-reaching effect. What actually is regulating the clonal hematopoiesis, the phenomenon itself? So if you think about about the model I initially depicted for you, that you have one mutation occurring in the expansion. And one thing you can say, well, why different people collected at same age group, some will have a very high level of clonal hematopoiesis, the clone size is very, very big. Some of them have very small clone size. One explanation say, oh, this is simply luck. Those ones who has a large clone tend to have it early, and those ones that have smaller clone may be acquiring it later, mutations later. So this is really the hypothesis on potentially the initiation stage being different in those groups. But there are some indications that this is not the only reason. And so if you think about the process, there's the initiation of the mutation acquisition process versus once you acquire the mutation, how well that mutant clone can expand. So this expansion regulation process, what are potentially regulating the clonal competition between mutants clones and wild-type clones, which has been very poorly understood process. So some evidence supporting that, for example, in this study, in this particular patient, for example, 2 time points collected, one at 56 years of age, another at 68 years of age. You can see that the allele frequency for this particular mutation is actually decreasing in the person, suggesting that not always. The fact that you can actually detect it in the starting with at the very beginning already says this is a dramatically expanded clone. Otherwise, you will never be able to detect it with deep sequencing. But yet, with age, further more than 10 years, actually started decreasing, suggesting there's a very complex process to regulate the clonal expansion process. So there are some known factors to be associated with the process and the severity of the clonal hematopoiesis. One of them is aging. Is smoking. So if you think about it, it could be a very simple explanation that smoking will lead to lots of potential chemicals in the bloodstream that actually leads to increased mutational rate. So basically that can be the cause of initiation of the clonal hematopoiesis. But you can also imagine that because smoking is also associated with inflammation, especially in lung and other places, there may be actually also potentially regulating the expansion of the clones through inflammatory regulation as well. The second thing, actually, there has been some studies noticing some links between solid cancer presence, as well as the clonal hematopoiesis. The very first paper is a Nature paper in 2013, where they discovered one of the genes involved in clonal hematopoiesis. At that time, they didn't call it clonal hematopoiesis, they called it mosaic PPM1D mutations in the blood, is associated with breast cancer and ovarian cancer. So this is a gene that's a phosphatase that's known to dephosphorylate PPD3 pathway, especially PPD3 itself. So, and it's a gene that's being observed in clonal hematopoiesis to start with. So that's one evidence. The second evidence is a recent paper published in Cell Stem Cell, where they noticed that there's therapy-related relationship between therapy for cancer therapies as well as clonal hematopoiesis. So that really ask the question, is this the cause or consequence or both of them? Right. So I would imagine that some of them are definitely cause. For example, cancer therapies will be treated with chemotherapies many times, or irradiation therapies, and those therapies could be the source of the mutations you actually observe clonal hematopoiesis. But it also could be serving as a consequence as well beyond just the causes. So I think there are more things to look into. And lastly, there's one very interesting aspect that we are very interested to think about. So that relates to the fact that once you have a clonal hematopoiesis, you have increased rate, well, if you have a higher allele burden, you have increased rate of progression towards some form of hematopoietic malignancies. But the rate overall is very, very small. So basically, you have less than 5% chance, even though the initial clonal burden is very, very high. So one possibility is, of course, you need lots of mutation to acquire to get any form of this, so that takes time to get that. But however, we know that in the case of leukemia, especially acute myeloid leukemia, you don't need many recurrent mutations. So in the TCGA data set, if you look at acute myeloid leukemia, on average, you only need 5 recurrent mutated genes to be associated with acute myeloid leukemia. And in some subforms, you only need 2 genes to be mutated to be associated with acute myeloid leukemia. And why, you already have one of them here in the clonal hematopoiesis, why the progression is very, very slow. So potentially there's a big bottleneck here naturally built into the system to prevent transformation and eventually leukemia development. And that's something that's clearly a black box currently, and we have no clue what's regulating it. If we can understand what's here, potentially can unleash some new passes towards safeguarding against cancer. So with this, I want to thank everyone who has been really contributing. In my lab, it's Wan Pen who has been championing the whole project, and we got lots of help from Marcus, without whom we cannot do this work, and as well as Richard Flavel's lab, and we got funding from the skin spore pilots, without which we also wouldn't have enough money to work on this process. And we have been working with Sarge Khan on solid cancer, on colon cancer on this front now, and also Kim Bleman to look at some pathology as well. Thank you very much.
Any questions offhand? So maybe I'll start while people are thinking. I think it's just disconcerting that as one ages that almost everyone has clonal hematopoiesis, and it's up to like 30% of the bone marrow, as I understand it. The DNMT3A allele, is that always the 882 codon, or is it spread out, or mostly the 882 codon like it is in AML, or is it other locations as well?
Yeah, so the